Today we are going to discuss a practical, and this is the 9700 paper 3-1, which was examined in June 2020, which exam was never held because the Cambridge went for the expected grades. So we start with the question. The first question, which was about, let's start reading the question right from the top, and that is the normal procedure that we follow. Before you proceed, read carefully the whole of question 1 and question 2. Plan the use of the 2 hours to make sure that you finish the whole of question 1 and the whole of question 2. So which means that you have to read the whole question so that you can understand the whole question and then decide whether you want to repeat the proceedings, or do you want to do it again, or you don't want to do it again. Now we start off with the information that is given to us. Invertase is an enzyme found in honey. Invertase breaks down sucrose. Now you know sucrose is a disaccharide. It is made up of a glucose and a fructose joined together. So it is a disaccharide. Into reducing sugar. So naturally, if we break down sucrose into glucose. The sucrose will break down to glucose plus fructose. Invertase breaks down sucrose into reducing sugar as shown in figure 1.1, and this is the figure 1.1. So sucrose reducing sugars, and I've told you the name of the reducing sugars is glucose and fructose. You will carry out investigations to determine the concentration of invertase in a sample of honey. The presence of the reducing sugar can be detected by using Benedict's solution, and you know Benedict's solution is blue in color. The time taken for the Benedict's to first show a color change, which means that as soon as its color changes from anything else rather than the blue that it originally was, so it is a very lightish blue, and whatever color it changes to, whether it changes to a green, whether it changes to a red, that is the first color change which you have to note down. So you have to note down the time taken for the first color change. As soon as it's no longer blue, you note the time, and that could be 10 seconds, that could be 20 seconds, that could be 30 seconds. Now you know that more the reducing sugar, now let's think of it theoretically, more the reducing sugar, the earlier it will change. More the reducing sugar, the lesser time it will take for the first color change. So the presence of the reducing sugar can be detected. The presence of the reducing sugar can be detected by using Benedict's solution, and the time taken for the Benedict's solution to first show a color change can be used to estimate the concentration of invertase in the honey extract H. So we've been given the honey extract, so there will be a beaker on which we will have labeled H, and we will have made this fake honey on the instructions what Cambridge would have sent us. Then it says you will need to prepare a serial dilution of 1% invertase. Now naturally all the other serial dilutions have to be less than 1. Can't be more than 1. You have 1% invertase solution E. So you're going to make something less than E. You are provided with the material shown in table 1.1. Okay, now we look at the table 1.1, what is shown to us. Now when we scroll down the page, now we look at table 1.1, and table 1.1 is what? H is the honey extract which we made. There's no hazard. And look at the volume given to you, 10 cm cube. This must have been instructed by Cambridge to give you this amount. E is the 1%. So the beaker labeled E is the 1% invertase. It's harmful, it's an irritant, because enzymes, if they touch your skin, they could damage your skin. And that's again 10 cm cube. B is the Benedict solution, it's 30. S is the 5% sucrose, and this is 25. Always look at the volume, because you should have used most of the volume. And W is the distilled water, that's fine. Then it says, if any solution comes into contact with your skin, wash off immediately under cold water. It is recommended that you wear suitable eye protection. Think about the hazards of using the invertase solution E as shown in table 1.1. State whether the risk of using the invertase solution E is low, medium or high, and give a reason for your answer. Now you could have come up with anything. It's medium was correct. You could have said high as well, because it's an enzyme, and what could the enzyme do is, of course, if you could spill it on your face or on your skin, it could damage the skin. And if it by mistake falls on your eyes, of course, that's why you're supposed to be using eye protection. So medium or high risk, and the reason is that the enzyme could result in some sort of, if it spills on your hand or something, well then of course it could create problems. Now as we could proceed along with the question, we're supposed to read the whole question. You need to carry out a serial dilution of the invertase solution E. Now if you remember E is a 1% invertase. E is a 1% invertase. To reduce the concentration by a factor of 10. So it's a serial dilution, and you need to reduce by factor by 10 between each successive dilutions. Then you have the next figure where they want you to see, they prepare the 1% and 0.1 concentration of the invertase solution. Complete figure 1.2 by drawing as many extra beakers as you need for your serial dilution. For each beaker, state under the beaker the volume and concentration of invertase available for use in the investigation. Then use one arrow with a label above the beaker to show the volume and concentration of invertase solution added to prepare the concentration. Use another arrow with the label above the beaker to show the volume of W added. So you should very carefully read this question and underline the keywords, and then proceed as you go along. Now look at this diagram very carefully, and it's very self-explanatory. As you look at it, it's not something very difficult. You see it has 10 centimeter of 1% invertase solution, and that was E. And we added that. So this arrow is shown to you. So we added 10 of 1%. And then we took out 1 out of it. So this is the one that we've taken out of it. What is remaining at the back is 9 cm cube. So 10 minus 1. This is the one. So 9 cm cube of E is left behind to use. So 1 we have taken out, and then we've added 9 cm cube of distilled water. So to the 1 we have added 9 cm cube. Now this is a total of 10. Now this volume, this whole thing should be 10. But no, it says 9 cm of 0.1% to use, because this was 1%, and 1 divided by 10 is 0.1%. So this is 0.1. Or we had to reduce it by 10 times. Now you can see how we have taken out 1 cm cube. So 1 cm cube of 0.1%. So we've taken that out and put it in another beaker. So this would be the third beaker. Now you can see how the 10 times it is decreasing. So 1% became 0.1%. Now this will become 0.01%. And this one will be 0.001%. And the next one will be 0.0001%. So what we need to understand is that we are diluting it 10 times, and every time we are adding 9 cm cube of distilled water. So every time we have 9 cm cube left, because every time we are taking out 1 cm cube, and we're adding it here, and we've added 9 cm cube of water. So the next we're going to take another again out, 1 cm cube, again we've taken out, and we've added 9 cm cube of distilled water. So this is a very simple way of doing a dilution. But this was the first two beakers here, were the one which gave you the clue of how you were going to do it. And if you look at the first two beakers, they guide you as to how you're going to make the next three beakers. Because the first two beakers told you that, okay, you added 9 cm cube every time. You had 10 cm cube initially, but 9 cm cube was left. You took out 1 cm cube, and then it was mentioned here 9 cm cube of distilled water had to be added each time. I mean, you can see how every time 9 cm cube of distilled water has been added. Now if you look at this table, this was for how many marks? This was for 3 marks. So we had to mark you out of 3 marks for this. And the 3 marks was label under correct sequence of beakers. So if you had given me this, if you had given me 0.01, if you had given me 0.001, and then you had given me 0.0001, and you had given me percentage, well, you got 1 mark. Then if you showed me that you had transferred 1 centimeter each time, like here you can see we have transferred 1 centimeter each time. This transfer is the one which you had shown here and then here again. So if you had shown me that, well, you got another mark for that. 1 centimeter, if you showed me transferring each time, you got another mark for that. And then if you had shown me 9 centimeter of cube of water, 9 centimeter cube of water being added each time, well, you got another mark. Then as we read along the next page, it says carry out step 1 to step 14 to determine the concentration of invertase in the honey extract H. First you have to prepare the invertase as you had described earlier on, and use a glass rod to mix the invertase solution and water. Set up a water bath, heat 30 degrees Celsius. Label test tubes with a concentration of invertase solution prepared in step 1. Put 2 cm cube. So we put 2 cm cube of each concentration of invertase solution into appropriately labeled test tube. Then you label another test tube H. Put 2 cm cube of the honey extract H into the test tube, labeled H. Then put 2 cm cube of S into each labeled test tube, including the test tube labeled H. Let's go through this carry out steps, page 6 once again. If you have a hard copy of this practical, you can see what I'm reading. Carry out step 1 to step 14 to determine the concentration of invertase in the honey extract H. So we have H, which I've shown you here in red. And it says prepare the concentrations of invertase solution as you described in A3 and shown in figure 1.2. Use a glass rod to mix. So we've got a 1%, 0.1%, 0.01, 0.001 and 0.0001% of the invertase, which is an enzyme. Now keep on thinking of the theory when you are actually planning this whole experiment. Set up a water bath and heat the water to 30 degrees Celsius. Now you know it's an enzyme, so we're going to keep it at 30 degrees Celsius, which is sort of an optimum of most enzymes. Then you label the test tubes with the concentrations that I've just done. So 1%, you've labeled all these. Of course, you label them on the test tube, not like what I've done it at the bottom of it. Then you put 2 centimeter of each. So we have added 2 centimeter of the different concentrations. So here we have the 1%, then we have the 0.1, and then we have the 0.01. So it's all the same volume, but the concentrations have changed. So 2 cm of each concentration into the appropriately labeled test tubes. Then label another test tube H. So we've got another one which is H. So we've got 5 test tubes, and we've got a 6th one which is H. And you put in 2 centimeter of the H in it. So the honey extract, which of course also contains invertase, and we want to find out how much invertase is in the H. So this is the question mark. Then we have put 2 centimeter of honey H into the test tube labeled H, then put 2 centimeter of S into each. Now what was S? Now if you look at the first page, S was the 5% sucrose solution, because we've got invertase here. The enzyme is invertase, which breaks down sucrose, and we want to find out how much invertase is there in the honey. So we're going to add sucrose to all of them. So this is the sucrose that we're adding, which I'm shading in green, and we're adding the same volume of sucrose in each, and this is 2 cm cube. So we've added 2 cm cube of sucrose into each test tube, including the one which is H. Now we've added it. Now you can realize now the reaction is starting, because as soon as we add it, the reaction has begun, and this is what there's an error in this experiment. So put all of the test tubes into the water bath at approximately 30. Leave the test tubes into the water bath for 10 minutes. So, okay, we've left them in 10 minutes, but realize this is that we mix this first and this a little later. So the reaction started in this, and then we mix this and this, and so the reactions have all started, and we're putting it in the water bath for 10 minutes. Now keep on thinking of what we're going to be talking about, the errors. While you're waiting, use your time to continue with question 1. After 10 minutes, remove the test tubes from the water bath and put them in a test tube rack. So you take them all out and you put them in a rack, all of these test tubes. So you put all the test tube in a rack, which is now, you've taken them out of the water bath, so now they're becoming cold and they're attaining the room temperature. Now after that, you've added 4 centimeter of Benedict solution. So we've added 4 centimeter cube of Benedict solution in each. So 4 centimeter of Benedicts in each. Now you realize the Benedicts have started to react as well. Now heat the water bath to boiling. So now you've increased the temperature to 100 degrees Celsius. Now it is boiling, and then you stop heating. And the first thing you do is, in the water bath, you remove all these other which you put in a rack first. Now you take a water bath, and you have this water bath at 100 degrees Celsius, and you put this test tube H in it, and you know the H had the honey in it, the 2 cm cube of sucrose in it, and the Benedicts in it. And you have now started to time. Now as soon as you start timing, you put the stopwatch on and you wait for it to change color. So measure the time taken for the first color change. So as soon as the color is no longer blue, you see the Benedicts is blue. So as soon as it changes color, you're going to note that time. And it says that if it's not changed, there is no color change after 180 seconds, then you just stop it. So 180 seconds is the maximum that you're going to look at it. And if it doesn't color change, you just write more than 180. And so you've done this with the honey. Now you need to be putting all this one. So you're going to put this one and test it, then this one and test it. So you're going to do this separately. Now you realize this is all taking a little time. And then you're going to do this one, and you're going to put these in the water bath at 100 degrees Celsius. And then you're going to wait for note the time for the first color change. Now it says state the results for H. Now the results for H could be anything. Let's say it is 60 seconds. Could be anything. It actually doesn't matter as long as you just give me a figure. So let's say 60 seconds, and we wrote, because seconds is given, the units is given, so you just have to write a number here, a figure here. Now you make a table, and it's always very important that you give me the independent variable in the first column. So heading for the independent variable is E, or you could have said invertase, and you could have said concentration, and of course the unit must come in the heading. So the unit, which is the percentage, must come in the heading. You must not write it anywhere here. You must never write it here in each column. So we've said 1%, 0.1, 0.01, and so we've got 0.1. Now you can see in the table we have the different concentrations. I think I made a little error earlier. So 1%, 0.1, 0.01, 0.001, and 0.0001. Now time for the first color change, I mean there could be anything. I'm just cooking up the results. So say it took 40. Now naturally you've got to think of it theoretically. The more the concentration of the enzyme, the first color change will be dependent on the concentration of enzyme will work faster. More the enzyme it will work faster, and it will break down the sucrose. So it will do a quick color change. So 40, so the 1 would be the highest, shortest time would be for the 1%. And then of course we can come up with something else, like, I'm just thinking of all this, what could happen. So this could be, maybe this could be even 65, and this could be 70, and this could be 80. So record your results in an appropriate table. Now this was for 5 marks. Now what are the 5 marks for? Number 1, if you gave me this heading right, this was 1 mark. If you gave me this heading right, this was the 2nd mark. Then readings for at least 4 concentration got you the 3rd mark. And the 4th mark was the shortest time should have been for the 1%. So this should have been the shortest time. So you got a mark for that. And records the results to the nearest second. So instead of giving me fractions, please give me whole seconds. And that is how you have to give it to me in this table. So you got your 5 marks if you gave it to me. So 5 marks, number 1 for this heading, the first heading, the second heading, 2nd mark, and then as long as you gave me 4 readings, and that was the 5 marks. Now let's look at the question as we go ahead. It says use your results from A3. Now in A3, if you remember, well, we said 60 seconds. So now if you look at 60 seconds and you look at this table, 60 seconds was for 0.1. If you look at this, 60 seconds was for 0.1. So the concentration will be 0.1. Of course, this would be corrected according to whatever the student's readings were. So it's not going to be one answer for it. It could be any answer, but any answer would be correct. Then it says suggest how you would make improvements. Now this is the important part. Suggest how you would make improvements to this investigation in order to obtain a more accurate estimate of the invertase concentration in H. The part 6 which says when you want to make improvements. Now of course when you come and estimate, so then it's not that that's a very casual estimate, so you should take a narrower range on either side of the estimated concentration. We think it's 0.1, so we should take more between 0.1 and 0.2 and even before 0.1, so between 0.05 and 0.2. Then this is of course something which you write nearly in every one, repeat, so that we calculate the mean and we can remove the anomalies. Then you needed to maintain the water bath at 30. We took them out of the water bath. I think we needed to maintain them at 30. Then the fact that honey, though we are trying to calculate the invertase in it, but it could contain either sucrose or glucose of its own, because honey is usually sweet. Then of course the staggered start. What does that mean? That means is that you complete one and then you do the other one, because if you have mixed the enzyme will start working and you're going to treat you've mixed all the enzymes in the 5 test tubes, but you're not testing each one of them. So you should actually do one test tube at a time. That's called a staggered start. I mean, you start with one and then a little later you add the other, so that by the time you've come to that, then the 10 minutes of that have been completed. So these were the improvements that we usually talk about. Then part B of the question is usually a graph, and it says honey contains a mixture of sugars. The percentage concentration of the sugar changes over time. The percentage concentration of sucrose in honey was measured by for a period of 25 weeks. And the results are shown in table 1.2. Now this is a very important part of the practical, and nobody should be actually losing marks in this. Now what we have to understand is that always the first column in the table is always the independent variable, and that is always plotted on the x-axis. The first column is always the independent variable which we have changed, and that is always plotted on the x-axis. And this, the percentage concentration of sucrose, was what you measured, and this is called the dependent variable, and this is the result, and this is always plotted on the y-axis. So you can see the mnemonic for it is dry, and the mnemonic for this is fix. Now you have to plot a graph of the data. Now that's a very simple way that you have to calculate. You look at the smallest figure, you look at the biggest figure, you look at the smallest figure here, which was 0.5, and the largest figure was 2.3. Now you have to spread this data on this, on this graph. And the first and the foremost thing is that you need to first plot the graph, that you need to label the axis.